Yesterday, we talked about machine learning basics, and building onto that, we're going to discuss AlphaFold's architecture. And today, we're specifically talking about AlphaFold 2, which came out in 2021. And so let's dive right into protein structure predictions. So let's say that we have some protein sequence, and we're interested in figuring out what the protein structure is. Can we predict this, and with what accuracy can we do it? In what cases could we do a better job? And you can sort of see it just from the fact that we are interested in designing new sequences, whether that's with PyRosetta or different machine learning methods, that will be very useful to be able to go accurately from protein sequences to three-dimensional structure. So proteins, we know, are made up of a bunch of different amino acids. And so in the protein sequence, so this is a very nice slide where the sequence spells out data science. But if we look at the amino acids corresponding to each of these letters and sort of the structure that you come out of it, we know that from this primary structure, we are able to have some sense of what the secondary structure would be, especially if we recognize, let's say, cysteines or some type of helix breakers. But going from primary structure to secondary structure and then to what was shown here, the tertiary structure, and if you have multiple proteins that you're trying to put together, maybe the quaternary structure as well. And the goal is sort of to see what clues can be picked up at the sequence level that we can relate to secondary, tertiary, and quaternary structure. So formally, this is known as the protein folding problem. And when you worked on your basic folding algorithm, it is the exact same idea. So we have some energy well. We talked about the influence of entropy. We also talked about the idea being find the local minimum. And so in this case, the idea is always to find the lowest energy 3D conformation given amino acid sequence. So let's say that your search starts somewhere around here. You want to be able to minimize on the energy and then find the lowest energy conformation, which we expect to be well folded. And we also expect to be able to find this. And in physics-based modeling, we once again do this by defining some type of energy so that we can minimize directly. And in the previous days, you worked on Ref 15 as this energy score. And we also had constraints to sort of affect what was happening to the overall energy landscape. And now we're going to discuss how you would just go from straight some type of target sequence to the full 3D structure. So this slide is shown to explain sort of the previous idea and previous types of modeling protein structures, starting with homology modeling. So in this case, our idea is to have some type of template that we can rely on so that we can figure out, given this target sequence that has a structural similarity to a template structure, figure out some alignment, and based on that, figure out exactly the target model. And then with that, you want to maybe make some refinements and adjustments on the target model so that it has the best energy landscape. So in the early days, as you can see, this is from 2003, similar sequences are known to adopt similar folds. So in the earlier days, well, maybe if we can take the folds from the closest sequence as our template structure, we can sort of feed that in and inform what our target sequence should look like in 3D. And then essentially, once we get to this point, we can have some energy-based refinement on it and have the structure look a little bit more realistic, and maybe have some specific changes based on the target sequence alone. And so this type of modeling obviously is very well related to how much data we have, because the more PDB files we have access to, the more templates we can essentially do this with. And also, in order to have the template structure available, we need to be able to have a very accurate sequence search. So that doesn't sound too difficult. However, sometimes you're going to get some alignment in one region and not as much in the other. So you can see if you have a very long sequence, maybe a part of it is possible to have some lookup in the database and it has a template, but maybe there's a domain that isn't currently as well represented in template form. And so in general, the more data you have, the better. But are there sort of limitations to this? Well, of course, but at this point in time, it seemed to work well. And once again, identify any related structures based on similarity in sequences, and then select your templates, align your sequence as best as you can with the template structure here, and then you start building the protein model for the target using template information, and then maybe you have some protocol for minimization. So we talked about machine learning models going from some type of training input to predicted output that we compared to the real output and get some performance rate or accuracy at this point. And we didn't talk about it in the context of proteins, but if we think about our sequence, so the protein sequence as the input for a protein structure prediction model. So let's say this model here is for us to go from sequence to structure. Then we would input our protein sequence, and then hopefully we come out with what? Coordinates. So maybe you're going from sequences to coordinates in this model, and it's learning how to get the numerical values that are closest to giving you that 3D structure. And we talked about there being a loss function so that we can evaluate the error during model training, validation, as well as during testing. We're going to want to know its performance and how accurate. And we're not going to talk about this right now because we covered it yesterday, but with proteins, you're going to want to have a bunch of different loss terms. And in AlphaFold 2, there's a lot in place to keep track of structural violations because you want to learn at the basis of that. So you can think of ideal bond lengths and bond distance, ideal bond lengths and bond angles, and you want to be able to keep track of these types of physical violations. So you can imagine the loss function is very involved, and it includes all of these types of ways of getting different types of errors. And so going back to this point right here, we know sequence is going to be our input and 3D structure is going to be our output. Well, maybe these are coordinates. Let's talk about exactly how we might want to represent proteins so that they are possible to it's possible to plug them in for machine learning in general. So we need a learnable representation for structure. And starting from the sequence, so you can have protein sequence. In this case, this is the N-terminal, and then we have something here. So my question to you is, how do you think we should represent sequence? Okay, so let's come up with a number for every single amino acid type. What's this one? Okay, all together, everybody say the one-letter code. Everybody. Okay, well, you did get through it faster than the interns, so some points for that. Okay, so what if we came up with, just like you were saying, what if we came up with a way to represent everything here in terms of one number? So if we were to do that, now let's go through this and put it in this token form. So Okay, now we have this. Is there an advantage over having this blue form versus this green form? So maybe from this number or token representation, we can figure out a way to make it more meaningful for learning purposes. Here's the thing though. A is 1, C is 2. 1 and 2 are real numbers, and 1 and 2 are closer to each other than 1 and 20 are. Is there a reason A is made to be closer to C than A would be to Y? No. So are we artificially making things closer to one another for learning purposes and sort of messing up what pattern there would be naturally if we could actually just represent the chemistry of amino acids instead of adding arbitrary values and making them, closer or further? So that's a problem. What can we do about that? Okay, so what if we rearrange this by size? But then what other properties do they have? All right, so, well, what if we place them by size and charge? But what other properties do they have? Okay, what if we place them by charge and whether they're polar and hydrophobic and size and whatever else, but we can't do that. It's getting too complicated. That's also biased. So we're trying to eliminate as much bias as possible. And with your idea, were you thinking that the map has the string? Would you represent, let's say we have 3 residues and you made a 3 by 3, how would you actually represent the sequence in a pairwise map? Yeah, so we don't necessarily have a way of representing any differently than we would either with the amino acid lettering or with the numbering. So in here, we would still need either of those, right? So that might be a problem. But let's go back to this other idea, the one-hot encoding. And let's see. So this is actually shown here in the figure. This is the sequence one-hot encoded. But let's try to put it into some representation here. So let's say that I'm interested in one-hot encoding this sequence. What is this dimension? Okay, so we're hearing 20 by 1. This is 20. This is 1. For each of them. So then, what would this be? Okay. Let's do it. Let's do this. Let's say this is our first residue. So for the first residue, let's just make... Okay, so now there are 20 possibilities. But what is this? Which possibility is the first residue? So like that. And we're not going to do it for all of them, but what is the next one? This residue. Okay. And then if we do this, are we able to show any possible sequence in this representation? Does it work for everything? It standardizes to everything. Any problems with it? the more compact way has too much bias. So maybe that's a trade-off. Also, it turns out that that space isn't really a problem. So let's think through it. Let's say you're starting from scratch with a model that's given coordinates, and your goal is gets given a sequence, and your goal is get coordinates, right? So whenever it's given a sequence, we need some distinguishing pattern between that is based on this identity in terms of how it would influence the structure. So to get that influence on structure, we don't want to artificially influence based on numbers because your model is aware of numbers and can pick up a number-based pattern. But there isn't necessarily this pattern in amino acid sequences in nature. There isn't really, or in the biology of it, there isn't really this even spacing where things come after one another. So you're creating a hierarchy that doesn't exist. And if you feed that information, then you're also introducing a bias that is this numerical scheme. But if you do this, then it doesn't have that numbering. So it's no longer that, okay, C and D come after one another, because that doesn't have meaning. You're trying to, because this is a very complicated problem, you're trying to make it as fair as possible so that the patterns that you pick up are actually based on that sequence structure relationship and not your sequence representation structure relationship. Okay, let's talk about other types of information that you can have. So as an example, we talked about there being templates, we talked about sequences and sequence similarity. So we also can gather some evolutionary information from sequence alignment. So in here, we have a query sequence, and we're looking at its alignment to another sequence. And we see that in certain segments here, we have some alignment. And do you think that for those regions, we could figure out if there are, we can assume there are structural similarities between them, right? So that was the whole point of our previous discussion here, is that we can have this search and have some alignment that helps us start making at least initial guesses as to where the coordinates should be. Can you think of another type of information regarding proteins that we should represent? So we have sequence, we have some evolutionary information, maybe from sequence alignment. We never talked about structure. We talked about coordinates, that we want coordinates, but we haven't explicitly talked about representation. In the earlier days, we talked about other types of structural information, maybe how poses store structural details. Okay, so we know that there are secondary structure associated with regions of the sequence once again. So you have your helix residues in a protein that has, in this case, helical regions here, back to back, and then you're going to have beta sheets. You're also going to have loops. So if you could identify all of the secondary structures, do you think you could put it together in terms of tertiary contacts? Maybe. It certainly makes it easier, right? So if you could figure out a way to have secondary structure be some type of input, that would be helpful. And then we also talked about some distance maps yesterday. So this is just another example. So once again, this is symmetrical, and you have the total number of residues by total number of residues. And the idea is that you can show the distance between each of them for any atom. So in this case, it could be all the backbone atoms combined. So it could be an average. It could be just one of the backbone atoms. It could be beta carbon, alpha carbon. And then if they are closer together, can you make some assumptions as to what secondary structure those residues would belong to? Yes. You might even be able to make some tertiary interaction conclusions from this. And then we know that there is Ramachandran plot associated with secondary structure, and especially in terms of ideal torsion angles. So if we had access to torsion angles and we had presented those, maybe that's another way to, it's another type of input to learn from. And then we also partially talked about burial. So we talked about protein core versus surface. So if you could contain the surface information, that would certainly give you some idea as to both sequence identity and also entirety of the tertiary structure determines the surface. What about the surface could also inform us maybe from a chemical perspective? Is it hydrophobic on the surface? Okay. Is there a protein that is hydrophobic on the surface? Type of protein? Yeah. There's membrane proteins. Right. What about the surface that could be useful in terms of function besides hydrophobicity? So maybe you have some charge distribution, and then what happens? So maybe with the surface charges, we can also get a pattern of where you would bind on the surface, and have a way of showing exactly what the activity would look like, at least for the most part as we understand it. And we know about coordinates, so you could have the XYZ represented. That's somewhat more trivial. And also, you could construct a protein graph. So let's talk about that. Okay, graphs. Two components this graph is going to have. What are they? Nodes and edges. Okay, what are the nodes? Let's say that we have a nitrogen atom, and let's say that this node is the XYZ. And then let's also say let's say I have another nitrogen atom and its own XYZ. Okay, so I have two nodes. What are the edges? The distance between them? So would this be something like that? Maybe the Euclidean distance between the coordinates of this versus that? Okay, so this would be an atom level graph. What if we wanted to make one at the residue scale? What are the nodes then? What's your favorite amino acid? Somebody? Favorite residue type? What's its code? Okay, someone else. What's your favorite amino acid? Quick. Somebody. Which one? Okay, so maybe I have two different residues, and then between them, we still take a difference to make the edges, you think? Maybe you could have interaction here. So distance is an idea, Euclidean distance. Maybe it could be some type of interaction. What's an interaction they would have? Could they be covalently bonded? Bond length? So that would be, but what type of bond? Okay, so maybe they could be making a hydrogen bond. Anybody else in the back? Right, interesting. So now we're talking about neighbors. So maybe there are other nodes around here, and then maybe there are neighboring interactions, and maybe there's a distance cut off to make it into the neighborhood. So that's interesting. Yeah, we're going to think about this as the basis of structural representation and how far apart everything ends up being and how learnable it is on the basis of graphs. So in terms of protein structure prediction, do you think it's more important to represent every single atom as a node or every single residue as a node? Residue. Do you think you need the atom-specific coordinate prediction? So if you coarsen to the level of residues, are you able to get a full idea of the structure? Do you think it's easier to get to this point and then maybe figure out a way to predict all atoms once you've placed the residues at a certain position? Is there a computational cost to doing everything at the atomic level? So maybe, once again, we're getting it to the point, it becomes problem specific. Maybe you're working on a problem where you can't not consider all atom interactions at all times, and you have to construct that graph. Can you think of such an example? So in fragment-based drug design, people tend to have to keep track of every single thing, including the binding site. So they consider all atoms at the binding site of a protein, and maybe some type of drug that's interacting with that binding site, as well as if they're trying to build the rest of the drug, then you need an idea of exactly where every single atom is at any point, so that you really obtain those distances. But also in general, do we have a way of perhaps having some type of penalty for breaking ideal bond lengths and angles? So what would that be in machine learning? So maybe it is okay to compute at the level of residues, but then have some loss for whenever your all atom prediction is too far off from the ideal bond lengths and angles. And so it turns out that's an effective way of doing this and approaching a number of problems in protein engineering. All right, so let's keep going. And we're going to introduce AlphaFold and talk about the entire model architecture. And before we do that, we'll just walk through multiple sequence alignment. So raise your hand if you've come across MSAs before. Okay, and if you haven't, earlier we had this figure right here showing that having some type of alignment in the sequence could perhaps reveal some structural similarity, and so maybe we want to learn from that type of data. And in here, this is a very abstract representation of a human protein sequence. So let's say that we take this human sequence and search against a database of a bunch of sequences from different types of species and figure out that there are these 4 other species that we were able to find, and they have some type of alignment to the human sequence. And so we line them up and see if we can learn from any sequence similarity to make structural sort of relationship to this alignment. So some way of getting structural information. And in here, the colors represent an amino acid type at that position for the alignment. And so what do we see here at the green in a very abstract sense? Well, maybe all of these, so this whole region, they're the same type of residue, okay? And then what do we see here in the middle? We see some type of crisscross. So what is that? What does that tell us? So maybe it's still conserved, but there's a little bit of flexibility. Is there a similarity between how this crisscross happens in the alignment? Yeah. Okay, so maybe this versus the reverse of it is informative in terms of some structural relationship. So maybe evolutionary speaking, we can relate the blue then yellow to the yellow then blue. So what do you think their structures would look like? This is going to be called coevolution signal. And what we see is that in terms of the structure that we have at this region, even though these 2 residues, the identities can sort of flip, their interaction is conserved because their interaction still occurs in the sequence at the same distance. However, they don't have to be at specifically, first position being yellow. It can be first position is blue, but since yellow is still there, they can keep that interaction. So from this type of signal, we can gather even more information for protein structure in general, and then put everything together to come up with 3D representation. And yeah, so we can make coevolution matrices, and we can also make conservation matrices. So in this case, a conserved region would be this, and using all of that, putting it together, we can come up with a structure and maybe something that's very learnable fundamentally for structure. And here is a coevolutionary weight matrix. So once again, our residues, so this is total number of residues. We're going from the N terminal to the C terminal. And the idea is that just based on evolutionary information, we could predict the contact so close to the ground truth. And this is somewhat what unlocked the power of using coevolutionary signal in machine learning. So this predates AlphaFold, and it was conducted by Sergei Ovchinnikov once again, and sort of leading into CASP. You've seen this figure before. We could see that there was a breakthrough, and based on so the success that was never achieved before, we could tell that this was a breakthrough in terms of our ability to predict structures, but also what got us from this point to this point. We're going to get into the entire AlphaFold architecture and try to understand exactly what it does to get to that highly advanced and highly accurate structure prediction. And maybe at this point we can just have a quick 5-minute break and then come back. We're going to dive right into it. Now we're ready to talk about the overall architecture and how exactly AlphaFold is able to reach very high accuracy in protein structure prediction. So we're going to analyze and sort of go through different components, try to put it together, and understand what's really happening in the two main modules of AlphaFold. So looking at this, this is it's a little bit more straightforward than looking straight at this figure. So what we're going to do is call this our sequence. So once again, it's the same abstract representation for a sequence, and we learned about multiple sequence alignment. So this is what the alignment would look like. And if we have sequence that we can search against database and it's possible to come up with some MSA, then we can go ahead and construct some type of a similarity matrix based off of the alignment and feed this through our network. But before we do that, I want to ask you if you can think of a case for where we have a query sequence and we search against database, but we don't really come up with that much alignment. So we don't have a very deep MSA. So what kind of a protein sequence would be hard to have a deep MSA search for? So maybe if the sequence was made by some lab, it was designed maybe de novo, it doesn't have any type of relation to other naturally occurring sequences, it might be difficult for us to find any similarity at all. Any other ideas? Or on the opposite end, what type of sequence would sort of result in the deepest MSA search? Okay, so what kind of protein, for example, what kind of protein sequence is very well aligned and there exist many things like it? Okay, well, in this case, these are protein sequences, but I mean, yeah, you could have similar problems in bioinformatics. Okay, so what about hemoglobin makes it possible to have this search? What in the sequence would align? Maybe what regions? Or why? Y hemoglobin. So maybe the heme group and the ability to recognize and bind such that other types of species had to do it a bunch of times is a function that has been sort of kept in nature and evolved a bunch of times and was too useful to get rid of, that there's a lot of examples of it, right? So maybe because of that, it's easier for us to come up with a very well-defined and diverse similarity matrix, and then exactly come up with the correct templates such that we can figure out, okay, there is so much alignment that the entire structure can be very coarsely defined by its similarity to these other sequences that are very similar, or these proteins structure aligned would make sense. And so we're going to have an MSA module dedicated to processing this type of information. And this MSA module learns at the basis of taking in this type of input and putting it in terms of MSA embeddings so that we can go from some initial coordinates, plug into our structure module and figure out the correct coordinates. And so one very important process, so very critical detail, is going to be called recycling. And so the idea is, well, we have some MSA, so we have some of our search here. We can figure out possibly some templates to go along with it. We can feed all of these into our MSA module, which will come up with some sequence embedding. And the sequence embedding itself will be useful in terms of figuring out the coordinates. But once we have our coordinates, or the first sort of initial prediction and then our final prediction, so we have one pass through the structure module and have some XYZ, what we can do is take this information and go back and recycle so that we informed by our previous prediction, we can figure out the correct embeddings until we can make this as accurate as possible. And so this was very successful in AlphaFold. And so given that we have a very deep MSA as opposed to very shallow MSA, we could figure out so much information from just this and use MSA module and a number of recycles until it would get quite accurate at getting to the correct XYZ. And so one other detail as to when we might stop recycling is another very important component to AlphaFold. So if it was you working on DeepMind's team and your goal was to figure out, okay, well, these are the coordinates to help inform the next prediction, so cycle through this, but use this to help you out as well, at what point would you know to stop recycling? What are some ideas? How many recycles should we do? Yeah. Okay, so maybe the answer would converge here. Okay, that's one idea. Okay, so maybe you have some metric to independently evaluate how good this XYZ is so that you know when to stop based on some score threshold. Okay, one more idea. Okay, so maybe a mix of both Donovan and Caroline's idea. You can sort of have both some convergence to the final XYZ, but also it's above a certain threshold you would consider it to be successful. And maybe if not, maybe you start over or something like that. Yeah, you could also have a certain number of recycles that tend to work for almost all protein structures. And maybe we have some way of sort of keeping track of what should be the default number. How do we know that? But let's talk about this part of the figure. So in here, what they're describing is, well, the output of structure module, which is the 3D structure of the protein, will have some regions that are high confidence and some that are low confidence. So what AlphaFold actually does is it predicts a confidence for every single residue. So this per residue confidence will be essentially that score that we defined. And so based on that, the user could identify, okay, well, this is very low in terms of the confidence, and I will have to repeat this a bunch of times until that confidence value increases to an acceptable amount. And in this case, blue is going to be high confidence. And so the more confident AlphaFold becomes in its prediction, it's possible to make this connection back to, well, how accurate is it, and given experimental results or in general ground truth structures, can we figure out the correct relationship, and at what point is confidence reliable versus not. So you'll get to do this very much hands-on, and you'll explore what happens with confidence at different regions, what type of sequence information is harder to, or what type of sequence segment is harder to have higher confidence on. And also, we'll discuss how recycling is injected back into the network, and at what point should you just consider either a very low number of recycles or maybe the very initial prediction. And here are some examples. So I wanted to talk about these two specific cases. We will dive more into it, but PLDT is the name of the confidence score we've been talking about. So this is a learned estimation of AlphaFold's own error. So AlphaFold learns how to estimate its own error. So whenever it makes a prediction for a structure, it also predicts how well it did that, right? So having that simultaneous training strategy allows for it to assess its own error, which unlocks a lot of power because you're able to learn sort of at multiple levels. One of the levels is obviously place the coordinates and make it as accurate as possible, but also learn how poorly you're doing the whole time. So there is a confidence head associated with, or let's just say there is capacity to learn what confidence is correct, and there is a loss associated with getting the confidence wrong as well as getting the structure wrong. So over the entire training epoch, not only do you figure out how to get the structure, you also figure out how to learn when you're doing wrong, which is very insightful. And so let's take a look at these two specific structures. So these are predicted. These are AlphaFold predictions of two different proteins. So two different sequences. You can see the lengths are different. They're both helices in secondary structure and some loop connecting helices. And we're going to refer to confidence as PLDT. So in this case, this stands for predicted local distance difference test. And there are other types of confidences. So AlphaFold doesn't only learn how to predict an LDDT, it also learns some other types, and we'll explore that. But this is sort of currently the most popular way of assessing confidence, and you may have already seen it in some papers. So once again, the color scheme is that blue is high confidence and red will be low confidence. In this case, this has a PLDT of 41.4, so this is very low confidence. And then this one is 83. So my question here is, what do you notice about these structures in general? So what is some observation? Obviously, their confidence is different. But let's focus on some maybe biophysical details here. Just looking at what the structures look like, what stands out. Maybe smaller sequence or a shorter sequence is easier to predict structure of. Let's test that today. So let's keep that in mind. Okay, let's try to get, so you don't have to focus too much on these numbers yet, but maybe just look at the structures. What's the structural difference between, yeah. So in this case, you have more contacts. Within this one protein, there are more contacts, whereas this one doesn't have it. Okay, what else? Which one makes more hydrogen bonds in this 3D, in its tertiary form? Maybe point. Okay, so we're thinking this one. So here's the thing. So these are the predicted structures, and this one's high confidence, this one's not. But I never showed you what the actual structure looks like for either of these sequences. So is it possible that this low confidence one is actually more accurate in terms of how well the structure aligns with the ground truth? And then this one with very relatively high confidence actually aligns very poorly with the real structure. So what we're going to do today is we're going to use RMSD and PyMol. So whenever you end up predicting an AlphaFold structure, you will compare it to the ground truth and make some conclusions based off of that and how the confidence changes and whether you can relate structure alignment to confidence, because that will tell you how well AlphaFold has learned to estimate its own error. So this, as well as obviously how well is it compared to the actual structure, will tell you about AlphaFold's performance. And what we'll do is to study, as Amy was suggesting earlier, to study some type of length relationship, we'll have people work on and study different proteins. So some of you will get a chance to choose a shorter one. Maybe everybody picks something different from each other so that you can compare. And then some people can work with longer proteins. Obviously, there will be a limit because, once again, the longer the sequence, the longer it takes for us to predict the structure. So there'll be a cut off as to the maximum number of residues you can do this with, so that you will finish in time. Also, you'll be using Colab GPUs for the first time, and I'm assuming everybody has the free trial, which at some point will run out of resources. So if that happens to you today, you'll have to make a new account so that we can continue with a bunch of other methods that also need GPUs on Colab. But let's take a look at this. So in here, we have PLDT, so this is confidence on the y-axis, and then we have residue positions. So our residue is 120 plus something. So let's say 125 long here, or we're analyzing basically from the 0th position to 125, and we're looking at the confidence, and then at the very top, we have the number of recycles. So the idea here was to take de novo protein and try to figure out how many recycles it takes to actually get it looking right. And the answer is very high. So in this case, until you got to 24 recycles, not only is it very low confidence here, colored by PLDDT, it also doesn't look right. So this is a beta barrel protein. And let's say we were to have a cutoff at recycle 3 and we stopped, we would very much have an unfolded protein. And so this is especially true of de novo sequences. What you'll find is that there's actually multiple papers on this by now, but for de novo proteins, people tend to have differences in terms of what number of recycles AlphaFold is run with, as well as what MSA information. So earlier, Chanel pointed out that, well, maybe for de novo proteins, there isn't as deep an MSA to do anything with. So maybe it's a waste of time to have that database search, because that also takes computational time. And so maybe you don't have MSAs and templates to go off of necessarily, and maybe the number of recycles will entirely depend on your problem and your protein. And if it's de novo, maybe this number is different. So in this case, we can see that it takes a longer time to get to this and more recycles. And then other things that vary the results of AlphaFold include the random seed. So you're able to change just the seed number and get different results, which is interesting, but also we know that there's a little bit of randomness in how AlphaFold is initialized, and so these can have real biological effects. So in this case, you're looking at this protein. So the query sequence of this protein is one with a knot in its 3D structure, and essentially having seed 0 versus seed 1 changes the outcome in terms of whether it has the knot or doesn't. And that's interesting. So these types of differences, especially for residues that have slightly flexible regions, for proteins that have slightly flexible regions, you might want to try different number of seeds and see if any of them are able to sort of randomly get a little bit more accurate. And so there is still the searching and scoring and sampling problem in AlphaFold and in the field in general. It just looks different with machine learning, and we've gotten to be sort of more accurate at what we do when we do score and do sample. So it's more guided by these methods, but there's still a little bit of searching that you have to do And again, depends on your problem. What we're going to do is we're going to talk through the AlphaFold notebook that is going to be a demonstration for AlphaFold. And we're also going to talk about the database search that is implemented in AlphaFold, as well as what settings you can change and what you got to predict. So we're going to get started with that. And this is the end of the lecture.